Hello, my dear fun beings. Sorry for making you all wait this long. In today's video, we will see the different models of plasma membrane. In our previous video, we have seen about the chemical composition of plasma membrane. If you haven't watched it, then the link is right here. Please click on it. Also, don't forget to hit the subscribe button and click the bell icon next to it. Now, without any further ado, let's get straight to the video. Before beginning with the models, we need to know that the plasma membrane is organized in an asymmetric fashion. This asymmetry in the membrane proteins, lipids and carbohydrates is based on the function that the particular cell performs. So, keeping this in mind, let's check out all the models of plasma membrane. To explain the biological and physical features of the cell membrane, there are two main categories of hypotheses. They are bilayer models and micellar model. Bilayer model are further of two types, lipid membrane model and protein-lipid protein or sandwich model. Let us see one by one. The lipid membrane model was proposed by Overton in 1902. He said that the plasma membrane is a thin layer of lipid because the fat-soluble substances can easily pass through it. Hoare and Fick found that the intact cell has low electrical conductivity, indicating a lipid bilayer. Gorter and Grendel said that the lipid bilayer is composed of polar heads facing the outer side. This paved the way for the future models. So, the next is the sandwich model. The sandwich model is further of four types: DANIELLI, DAVSON, unit membrane, greater membrane and fluid mosaic. Let us see one by one. The DANIELLI-DAVSON model. Harvey, Cole, Davson and Danielli indicated the existence of proteins in the plasma membrane. This led them to propose a lipid protein model. According to them, the plasma membrane has two layers of lipid molecules with their polar regions on the outside, and globular proteins are thought to be associated with the polar groups of this lipid. In 1954, they considered that these proteins are in the form of a folded beta sheet. They also suggested that the plasma membrane had pores which were lined by beta sheets of protein, here the red color, and the alpha helices lied on the top and bottom of the lipid bilayer, the blue color. So, this model remained popular for a long time. Next comes the unit membrane model. So in the unit membrane model proposed by Robertson, it was thought that the cell organelles had this kind of a structure. It is considered to be a trilaminar structure with a lipid bilayer sandwiched between two protein layers, totally making up 75 Armstrong. The proteins on the outside are mucoproteins, whereas the proteins on the inner surface are non-mucoid. That is, the protein layer is asymmetric. Both these models are examples of a non-typical membrane. The next membrane is an interesting membrane, which is the greater membrane model, also proposed by Robertson. He believed that the inner and outer surfaces of the membrane were asymmetrical and had structural polarity. The internal surface of the membrane were thought to be composed of unconjugated proteins and the outer surface with glycoprotein. So here, as you can see, the glycoproteins have oligosaccharide side chains with negatively charged sialic acid terminals, thus making the membrane asymmetrical and having structural polarity. Next, let's see about the fluid mosaic model. For this, we need to perform an experiment which requires the freeze-fractured replicas. Remember guys, freeze-fractured replica are required for studying membranous structures. So, while studying the freeze-fractured replica of plasma membrane under the electron microscope, the electron micrographs revealed the presence of many particles which remain embedded in the lipid layer. But when artificial lipid bilayer freeze fractures were studied, there were no such particles. When these artificial lipid layers were mixed with proteins, such particles are observed in its freeze-fractured replica, and these proteins penetrate into the lipid monolayer. It is also found that this is possible only if the membrane is fluid in nature. Singer and Nicholson proposed the fluid mosaic model, which is now widely accepted. This model is assumed as a continuous bilayer of phospholipid molecules in which globular proteins are embedded as discontinuous mosaic pattern. like icebergs floating in water. The integral proteins are arranged in a mosaic pattern, and the fluidity is due to the hydrophobic interactions of the proteins and lipids. These lipids exhibit flexion, lateral diffusion, flip-flop movement, and rotational movements. Next is the micellar model or subunit model. According to Moran and Sergestrand, the plasma membrane consists of globular units which are called elementary particles ranging about 40 to 70 Armstrong in diameter, which are packed together to form repeating units. According to Green, this consists of fused repeating units. Two types of membranes are distinguished: monopartite and multipartite. Monopartite membrane consists of repeating units which do not have any projections. For example, the membrane of RBCs and outer membrane of mitochondria. Multipartite consists of repeating units which form base, neck and head, and the base pieces are fused with each other. Example is inner membrane of mitochondria. It may be micropartite or macropartite. Now, let's see about the relationship between the bilayer and micellar hypothesis. So, there are evidences for the existence of both these hypotheses. That is, some membranes show the existence of bilayer hypothesis, whereas some show the existence of micellar hypothesis. Different membranes show different types of structures, and also there is a possibility of transformation between these two states. Hooray! With that, we come to an end of this video. Thanks a lot for watching, guys. And now don't forget to hit the subscribe button and click the bell icon next to it to be the first one to get your concepts right.